Hi, dear students. This is Rethik, sir, your biology subject matter expert from memoneet. So, yeah. So, I'll just make it a very simple that we have very small time for our preparation. So, that's why I'm here coming with some simple solution that is theory aspects, which is very important. Because, we keep on revising a number of MCQs for sure, but without being strong with theory. So in biology, if you're not strong in theory, then it has no use because you will not be able to understand the questions properly. It could be, like it could be a match the following or the assertion and reasoning type or the statement base, fill in the blanks or direct questions, anything. So without understanding or without having a grip or knowledge of theory, you will be unable to or you will be failed to answer the questions properly. So we'll go with some interesting, simple, yet very weightage, chapters. Okay. So we'll just try to complete as soon as possible. So the chapter is principle of inheritance and variations. So in this chapter, we will be covering the Mendele's law of inheritance, then inheritance of one gene, inheritance of two gene, sex determination, and the mutation. Genetic disorders are again a separate concept. So mutation and genetic disorder, I'll try to complete it in another video. So right now, because questions are being asked more in these as well as this part. Okay. So this part and this part. So questions will be asked more separately. So we will just try to concentrate and complete those parts separately. Right. So my major question is, have you ever wondered why an elephant always give birth only to the baby elephant and not some other animal? First question. So you'll always be thinking. And when it comes to plants, say like we have a mango plant here. So why a mango seed forms only a mango plant and not any other plant? A big question mark, right? So given that they do, so offsprings are also identical to their parents, or they may show somewhat variation. Okay. Like you have seen your siblings sometimes look so similar to each other. No. Or sometimes even different, sometimes very similar when it comes to like their twins, right? So there are several questions that we deal up with scientifically in a branch of biology, which is actually called as genetics. So finding solutions, finding answer to such questions in the branch of biology is actually called as genetics. Okay. So this subject deals with the inheritance as well as the variation of the characters from parents to offspring. Okay. So therefore, we need to remember here that inheritance is actually a process by which the characters are being passed on from parents to their progeny, that means parents to their children's. It is the basis of the inheritance, and variation is the degree by which the progeny differs from their parents. So remember, inheritance hereditary is something that has been passed on from the parents to their progeny. It is called as hereditary. Okay. And when it comes to what do you say that variations is the degree by which the progeny differs from their parents. So yeah, humans knew from as early as 8,000 to 10,000 BC, that is before Christ, that one of the cause of the variation was hidden in the sexual reproduction. Remember this thing. Okay, so they explored the variations that were naturally present in the wild population of plants and animals to selectively breed and select for organisms that possessed a desired character. For example, through artificial selection and domestication from the ancestral wild cow, which in India, very well known breed, that is Sahiwal cow, which is in Punjab. We must, so that means before only, even before the introduction or introducing of this artificial insemination or cross breeding, before scientifically introducing it, our ancestor knew this, but not scientifically, but they used to follow it. And this is one of the example for that. That is what they're saying. Okay. Now, coming to this part. where Mendel's law of inheritance, which is very important. So, what does this say? It was during the mid 19th century that this person, which is George Mendeleev, he conducted a set of hybridized experiment. Okay, so choosing the garden pea plant for several years and proposed the law of inheritance in the living organism. So during Mendeleev investigation, okay, so during his investigation into the hereditary pattern, it was the first time the statistical analysis, mathematical logic were applied to the problems in the biology. Till that, biology is just a theoretical concept, no mathematics, nothing. Very first time the Mendeleev, he has proposed, applied the statistical analysis, then mathematical logic, okay, were applied to problems in the biology. So his experiments has larger sampling size, which gave a greater credibility to the data. So if the sampling size was large and with you know doing repeating the experiment again and again, he was getting a consistent value. That means it has a greater credibility that he has collected and also confirmed of his inferences from experiment on successive generation of his test plant from one generation to the other generation. He confirmed whether the result is coming same or not, and that proved his result pointed to general rules that we got, that is rules of inheritance rather than being unsuitabilized idea. Okay, so what are all the things that came with it? See, Mendeleev, first thing it is, he conducted an artificial pollination. So, how many of you remember the pollination, sexual reproduction in flowering plants? If you do remember it, you have already came up with this, right? That is artificial pollination. So, what does this artificial pollination denotes? A cross pollination. Artificially, you are, yes, breeding or pollinating the plant. So is also like cross-pollination experiment using several true breeding pea lines. Pea lines are nothing but pea plants. True means original, they're wild type. A true breeding line is one that having undergo continuous self-pollination. Getting? So it has not went with the pollination with some other plants. Rather, self-pollination show the stability of inheritance, trait inheritance, and expression for the several generation. Once the character have not been divided or distributed, rather it has been passing from generation to generation. They are called as true breeds. Okay. So Mendeleev selected 14 true breed plants varieties as space, which was similar except for one character with constanting traits. Some of the constanting traits which were selected was smooth or winkle seed, one, then yellow or green seed, then full or constructed green or yellow pods, then tall or draft plant. Okay. So in this table, you are seeing the characters which he had chosen and their contrasting traits that you are seeing. Yes, this is very important because in the NEET, they can actually ask you as MCQs, match the following. In MCQs, they can actually ask you as match the following. Or reasoning kind of question might also come from this part. Next, coming to the inheritance of one gene. I hope you're seeing the picture here. So what does inheritance of one gene says that the Mendeleev, he crossed a tall draft pea plant, okay, to study inheritance of what? One gene. He collected a seeds produced as results of this cross and grew them to generate a plant of first hybrid generation. This generation is also called as filial F progeny or F1 generation. So Mendeleev observed that all the F1 generation progeny plants were tall, like one of its parent, none were draft. Draft means small. Okay. So as you can see, he has actually crossed them with the one true tall plant, as you can see here, and another one were wrapped. So what will be the gametes now? T and small t. So in F1 generation, all the plants were tall and not wrapped. So again, Mendelein, what he did is he self-pollinated it. So now he got this heterozygous gametes. So if you see it, again, you will have two gametes here, correct? So then he has self-pollinated it. So when he self-pollinated, right? So what he will get? Capital T and capital T, correct? Then capital T, small t, and capital T, small t, and small t. So what would be the ratio? The ratio will be 3 is to 1. So that is 3 will be the progeny expressed in the F2 generation will be 3 is to 1. The contracting trait do not show any blend in either F1 generation or F2 generation. So based on these observation, so 3 is to 1 in which 3 will be tall and 1 will be the dwarf. Okay. Out of 3, 1 is homozygous and 2 are heterozygous in condition. So based on the observation, Mendelein proposed that something was being stably passed down unchanged from parents to their offspring through the gametes over successive generation. So he called this thing as factor. Do remember this, okay? He called this as factor. Now we are actually calling this as genes. So genes, therefore, are the unit of inheritance. Okay. So they contain the information that is required to express a particular trait in an organism. So genes, which code for a pair of contrasting traits, are known as alleles. That is, they are slightly different from the same of the gene. So alphabetically, if you say symbol, then the capital letter is used for the trade expression of F1 stage and the small alphabets that you are seeing, right? For the other trait. For example, in case of characteristics of height, I'm talking capital T is used for tall trait, whereas small t that you are seeing, right? Simple. Alleles can be similar as in case of homozygous TT. Homozygous means similar, tall t, and small t, or can be dissimilar in case of, as I told, heterozygous, that is capital T and small t. Since from the observation that the recessive parental traits is expressed without any blending in the F2 generation, we can infer that when a tall and draft plant produces gametes by the process of what? Mitosis or meiosis? By the process of meiosis, the alleles of the parent or the alleles of the parental pair segregate or separate from each other, and only one allele is transmitted to the gamete. So this segregation of allele, that is separation of allele, is a random process. So that is 50% chances of the gametes containing either allele. Okay. As have been verified by the result of the crossing. Means only 50% chances will be there that the gamete will be inheriting both the parental character in their respective. So like in F2, F1 generation, what happened? All the plants were tall. But in F2 generation, what happened? The ratio was 1 is to 2 is to 1, right? So this was heterozygous. Here, these two, they are what? They are homozygous. And only this 50% was hetero. Clear? So, During fertilization, the two alleles, that is T from one parent says through the pollen and T from the other parents, then through the egg, are united to produce what a zygote that have one T allele and one small t allele. In other words, the hybrid have the capital T small t. That's what I said. Since these hybrids contains alleles which expresses the contrasting character, that is the character from both the parents. The plants are actually the plants, or that thing is actually called as heterozygous. Be clear with this, okay? And my dear, you need to know that the possibilities of gametes are written on two sides, usually the top row and the left column. Okay, so this thing you're seeing, correct? This thing is actually called as what? It is called as the Punnett square. So it was developed by a British genetist. So his name is Reginald C. Punnett. So the name came like Punnett. Okay, so it is a graphical representation to calculate the probability of the possible genotype of the offspring in the genetic cross. The possible genotypes are written on two sides. Usually, one will be like top and the left column. Say, like, I'm going to take, so if, okay, say you have like capital T, small t, capital T, and small t. Okay, sorry. Oh, this is heterozygous, right? Okay, right. Say like this is male and this is female. So we have capital T and the capital T. So we have small t and the small t. It is something written, okay? So don't get confused. I hope. So what will be the result now? So capital T, small t, then capital T, small t, right? So capital T, small t, and capital T, small t. That means in the F1 generation, all the plants are tall only, but they are heterozygous. That means they are hybrid plants. Okay. So how do you see this in F2 generation? Same thing will happen. So that is what they have given here. So when you have a gamete, selfing they will do. When they do have a gametes, right? So when that happens, like what is selfing here? So one is you're going to cross it with the uh here itself it is. So when you are going to cross it with the tall gametes, capital T and small t, and again you're going to cross with this again capital T and small t. So can I write here? So if you're going to cross with the capital T and small t, and again with the capital T and small t. So what you will get? Capital T, capital T, capital T, small t, then capital T, small t, and small t, small t. So that means here T, T, and small t, they are homozygous in nature. That means they're the pure line. So where they're tall and dark. So that will be like one and one. So another part you're seeing, this part, it is heterozygous. So that means 50% will be in F2 generation will be heterozygous. That is again tall. So one thing is clear, out of the draft and out of the tall, the one which is dominant, will be expressing its character in all the generation. Where the recessive one will be, uh, it won't express unless until they are in homozygous condition. Remember, recessive will be able to express only when they are in homozygous condition, not when they are in heterozygous condition. This is only relevant to this experiment because in future, like in the next slide, we will discuss where there will be a mixture of characters will also appear, where sometimes both the alleles will be dominant to each other and both will express together, or sometimes both will show express and bring up a contrasting character. So that all thing we are going to study. So what will be the final ratio here? Means what would be the final ratio? It would be 3:1 ratio. That is phenotypic. But when it comes to genotypic, it is 1:2:1. I hope you're clear with this. Now, this is a diagram representing a test cross. Okay. So when, see, what same thing when it comes to color here. So this is the white is the recessive and the purple will be the what do you say that the dominating one. So when you cross the white with the purple, right? So as a result in F1 generation, all will be purple because purple color is dominant here. But when in F2 generation selfing is done, so you will see what the ratio half of the flower are violet and half of the flower will be purple. Like either 1:2:1 ratio or 1:1 ratio. Anyhow. So based on the observation on the monohybrid cross, Mendeleev proposed two general rules, okay, to consolidate his understanding on inheritance in the monohybrid cross. So today, this rule or as a principle is called as law of inheritance, which is nothing but the first law of dominance, and the second law or the law of segregation. So first, let us go to the law of dominance. That's what when I'm talking about test cross. Law of dominance, characteristics are controlled. Remember, so they can even ask this as reasoning or statement based question. Clear? So law of dominance, where the characters are controlled by the discrete units, they are called as factors. Good. Factors occur in pairs, obviously, because nowhere you're seeing it individually, either like this or like this or like this, right? So it will be occurred always in the pair. In a dissimilar pair of the factor, see, one member, a pair of will be the dominant and the other will be the recessive. So the law of dominance is used to explain the expression of one of the parental characters in a monohybrid cross in the F1 and expression of both in the F2. So it also explains the proportion of 3:1 obtained at the F2 generation. Based on this, this comes a law of segregation. So this law is actually based on the factor that the allele do not show any blend and that other characters, okay, recovered as such as in the F2 generation. Oh, one of this is not seen in F1 generation. So that is law of segregation. That means even though in F1 generation you won't see this any kind of character, but still in F2 generation you are going to see it. So how? I have a very good example. So, two the parents contain two allele during the gamete formation. The factors are allele of phase segmentations from each other such that the gamete receives only recessive only one of the two factor. Of course, homozygous parent produces all the gametes will be similar, while the heterozygous, two gametes will be different. Okay, each other one allele with equal proportion. So there comes a law of independent assortment. Okay, what does this law actually defines? Yes. So when an expression of a P was repeated using other traits in the character, it was found that something in the F1 had a phenotype that did not resemble either of the two parents between the two. So the inheritance of flower color is actually called as the dog flower, or it is also called as snapdragon, or it is also called as Antrinium species. Okay, so this is the different name, and this is the best example that I can give you. So what happened here is when a red plant was crossed with the white plant, white flower plant, so gametes, will be red and small r. So as a result, in F1 generation, all the plants were in pink, flowers were in pink. So it is something which you have not observed or which you have not seen in the F1 generation here. So that means it is neither resembling the first nor a parent. That's what. Means in between. Okay. So the inheritance of flower color in the dog flower, where incomplete dominance you will see. Okay. So when it was self-pollinated in F2 generation, as a result, what happened? You will see 1 is to 2 is to 1 ratio. That is red, which is homozygous, 2, which is pink here, and another one is white, that is 1. So genotypic ratio will again be 3 is to 1. No, it is 1 is to 2 is to 1 here. See, in the previous slide, I just, where it is here, I told you phenotype will be like 3 is to 1. But here you are seeing the phenotype is not 3 is to 1, rather it is 1 is to 2 is to 1. Genotype is 1 is to 2 is to 1, that is correct. So what exactly is the dominance then? So then what exactly is the dominance? And why are some alleles dominant and some are recessive is the big question that comes. So to tackle this, this was the experiment this done. And there is another example where the gene that contains information, it is, yeah. Let's take another gene that contains an information of producing an enzyme. Now there are two copies of this genes. Okay. That is two allelic form. Let us assume that the normal allele produces the normal enzyme that is needed for the transformation of the substrate. Yes. Therefore, the modified allele could be responsible for production of the normal, less efficient enzyme or a non-functional enzyme or no enzymes at all. So in first case, the modified allele is equivalent to the unmodified allele. That is, it will produce the same phenotypic trait that is result in the transformation of the substrate. Yes, such equivalent alleles are very common, but if the allele produces non-functional enzymes or no enzymes, then the phenotype might be affected. In second case, the phenotype traits will only be dependent on the functioning of the unmodified allele. So here the unmodified functional allele, which represents the original phenotype in the dominant allele, and the modified allele is generally the recessive allele. Hence, in the above examples that I have discussed, the recessive trait is seen due to non-functional enzymes or because no enzymes are produced. So this is again very important because this is called as codominance. So when till now we have discussed regarding the crossing where the F1 resembled either of their two parents or was in between. That is incomplete dominance. One is dominance, then incomplete dominance. This is what we have discussed. But in case of codominance, what happens? The F1 generation resembles both the parents here. Okay. So one ABO blood grouping are considered as one of the best example here. So the plasma membrane of the red blood cells has sugar polymers that protrude from its surface, and the kind of sugar is controlled by the gene. So the gene one has three alleles. One that is gene I. Okay. So it has three alleles. One is IA. So you can say like IA or IEB, and another one will be the small I. So allele IA, IB produce slightly different form of the sugar, while the allele small I does not produce any sugar itself. So because humans are diploid organisms, each person possesses any two or three kinds of IE gene allele. So if the parent one is having IA and the parent two is having IA, the offspring will be having what? IA, which means A type of blood group. And if from the parent one, if I get IA, what do you say that allele and IB allele from another parent. So then as a result, it is heterozygous. But here, instead of expressing something which is no another contrasting color or some new character or any one dominant, here both IA and IB are expressed together. So as a result, both the alleles are being expressed. So the offspring will be having A and B blood type, AB blood group. Now you say, what is another example? Yes, IB and IA. So this part is also another example. Coming to the recessive part, since so this is next concept will be inheritance of 2G. So coming to the inheritance of 2G, Mendelian also worked with and across pea plant that differs in two character as seen in this plant. So the character will be having round yellow and wrinkle green. Round is one character, yellow is one character, wrinkle is one character, green is another character. Okay. So now what happens here is in F1 generation, when a parents like round yellow character PE is being crossed with the wrinkle green. Okay. As a result, R and Y, R and small r and small y, capital R and capital Y will be the gametes. As a result, what you're going to get in F2 generation, it will be capital R, capital Y, small y. So when you do a selfing of it, so what you are going to get? Yes. One thing you will get a see, I'm just trying to write it here itself. Okay. So as a result, you will have four gametes here. Correct? Then, sorry, capital Y, small r, and small y. So here as a result, capital R, capital small r, then capital R, small y, then small r, and small r, small y. Correct? So is it correct? Yes, it is correct. Where it is R and Y. Got it? So no confusion. You'll have totally four gametes here. So as a result, see, like this is what I'm going to do. So capital R, small y, then small r, capital Y, then, okay, this is small y, right? Okay. Then capital small r, capital Y, small then capital Y, small y. So what you're saying, capital R and capital Y you're seeing, then small r, capital Y you're seeing, and capital R, small y you're seeing, then small r and small y you're seeing. Correct? Yes. So now, once it has been done, what is going to happen here is the crossing over will results in the phenotypic ratio that is 9 is to 3 is to 3 is to 1. Okay. So what would be the phenotypic ratio here? 9 is to 3 is to 3 is to 1. So what would be the genotypic ratio? Nothing, because all these are different now. So again, it will be 9 is to 3 is to 3 is to 1 only. So this is law of independent assortment. So what actually it says in law of inheritance of two, gene here, you can tell that which of the character base has been alleled here. Okay. So as a result, in the dihybrid cross, the phenotypic. Then, what does the law of independent assortment says here? In the dihybrid cross, the phenotype round yellow, round green, and wrinkled green appeared in the ratio 9 is to 3 is to 3 is to 1. Such a ratio was observed for several pairs of characters, okay, that Mendelian studied. So based on that observation, you can easily see the percentage here. Clear? So the ratio of the phenotype will be 9 is to 3 is to 3 is to 1. Because round yellow, when that has been crossed with the wrinkle green as a parent in the F1 generation, in F2 generation, you will get round yellow only. That means it is clear that here round yellow is the dominant. Okay. Sorry. That means round yellow, right? That what we are seeing is the dominant here. So in F2 generation, as you can see that here what happens once the gametes has been there, so gametes has been crossed again. So as a result, so you're seeing 9 will be yellow, round, then 3 will be the round green, 3 will be the wrinkle yellow, and 1 will be the wrinkle green. So phenotype will be 9 is to 3 is to 3 is to 1. Genotype will also be like 9 is to 3 is to 3 is to 1. So this is about the law of independent assortment. Okay. So in the next video, we will actually be discussing more about the chromosomal theory of inheritance. So once the chromosomal theory of inheritance has been done, we can also move on to the what do you say that the pedigree analysis, then sex determination, and the mutation.